So good morning to you all. Today I will start with my topic with genetic code. My topic is genetic code. You know all the informations of our phenotype. All the informations of our phenotype about our behavior, it is stored in DNA. So the genetic code, it is stored in DNA. It is the blueprint of life. Okay. So before going to genetic code, we must know the central dogma. We must know what is central dogma. Central dogma means, look at that. Look at the central dogma. Central dogma means the information present in DNA. It will pass from through transcription. It will be production of RNA. RNA will undergo translations. Through translation, again protein will be produced, and protein is responsible for all phenotype. Okay. So look at that, how genetic information has been flown. How genetic information is coming. It is from DNA through protein, through RNA to protein as well as phenotype. Protein will be responsible for all sort of phenotype through enzymes. Okay. Look at that. The DNA reproduced in the form of replication. DNA transmit the information through transcription and translation to the phenotype. The information is not that. So look at that. It is a one-way information. Information is coming from DNA. Information is coming from DNA to protein. It is not that protein will determine your genetic code. It is that the information is coming. The flow of information is one-way traffic. Flow of information is one-way traffic. Is it right? Because it is the DNA, from DNA to protein, RNA to protein. Except in certain viruses. Those viruses which have the RNA is the genetic material. Those viruses who have the RNA is the genetic material. Look at this. They reproduce. This RNA genetic material. They again produce DNA. RNA also produce the DNA. Those DNA, it is intermediate. Again from the intermediate DNA, information is coming from DNA to mRNA to protein. So this is the central dogma of molecular biology. So what is the central dogma of molecular biology? Information is coming only from DNA to protein or to the phenotype. It is not a reverse. It is not reverse. Whatever the information present in DNA or RNA, it will come from the two steps involved in transcription and translations. In these two steps, information will flow from DNA to protein. Or it is not the reverse way. It is not reverse. Remember, environment, the protein, that means the phenotype. Ultimately, protein is responsible for a phenotype. Phenotype cannot dictate that what is genetic material you have. Okay, phenotype cannot dictate. So this is the flow of information. Okay, remember that already we know that the genetic information is coming from top to bottom, DNA to RNA. DNA is mainly composed of all the 4 bases. Ultimately, the information present in DNA, sequence of the base present in DNA, or in specific sequence of the RNA, sequence present in mRNA after transcriptions, they will be translated into amino acids of the protein. So those number and bases, the sequence of the bases present in DNA or mRNA or RNA is known as codon. What is genetic code? Genetic code, it is the sequence of the base present in DNA or RNA. What is a genetic code? Genetic code is the base sequence of the bases present in DNA to be specifically present in RNA is referred as genetic code. Is it clear? What is genetic code? Sequence of the base present in DNA, more specifically in the RNA, is referred as genetic code. Again, you already know that genetic code will be coming to the cytoplasm during the protein synthesis through translations. During translation process, amino acids of the cytoplasms will be coming, will be carried to the protein synthesis site through tRNA. through tRNA, amino acids will be carried to the protein synthesis site based on the sequences. So when there is a complementarity between base sequence of the RNA and base sequence of DNA, there will be certain anticodons. Complementary base pairing of tRNA and mRNA is responsible for putting the correct amino acids in accordance to the correct genetic code. So whatever is base present in tRNA, it is referred as anticodons. Okay. So there are 20 amino acids. You remember that there are 20 amino acids. Those 20 amino acids, there are a set of codons which are responsive for 20 amino acids, and it is referred as codon dictionary. That all the bases genetic codes for 20 amino acids are referred as codon dictionary. Okay, these are codon dictionary. Look at this. This is the process. What is genetic code? The information present in DNA, which is coming from through DNA to mRNA, it is codons, and ultimately from DNA, it is the codon. Codons are presented in the form of mRNA through translations. Ultimately, genetic codon, it is present. Whatever the bases present in the codons, it is referred as bases. It is codons. These codons will be ultimately responsible for different proteins. Okay. So this is the flow of information. So genetic information, it is like that way. Now look at this. What is tRNA? So in a codon, so this is the mRNA molecule. This is the mRNA molecule. Based on, look at the mRNA molecule. Based on the mRNA molecule, tRNA, which is the carrier of amino acids during protein synthesis or translation, the base codon and whatever the bases present in tRNA, it must pair. Without pairing, there is no protein, there is no pairing. Okay, so these whatever the alternate complementary bases present in tRNA, they are referred as anticodon. What is anticodons? Complementary base pairs present in. Look at the difference. Where you will get the codon? Codon you will get in mRNA molecule. Anticodons you will get in tRNA molecules. Okay, tRNA molecule. The persons. Look at this. These are two persons which are responsible for detecting, identifying the genetic code. Theoretically, these are two persons, Francis Crick and Sidney Brenner. These are the two persons involved in discovery of the genetic code in theory wise. You know Crick, Watson and Crick. This is what one of the partner of DNA discovery was Francis Crick, and Sidney Brenner. 1960, actually no, it should be 64. These two persons, 61 and 64. It is not 54. Sidney Brenner and Francis Crick, they are the persons involved. It is not 54. It should be 64 because in 54 no DNA was discovered. Okay, they were the persons responsible for determination of the genetic code. Okay, through certain theoretical process. But final experiment was proved, it was given later on. What is the behavior of the genetic code? Okay. So this is the coding dictionary. You can remember, you can write it down if you want. If you want, you can write it down. Look at that. This is the coding dictionary. Okay. This is the coding dictionary which always we refer. Okay. So how the genetic codes were discovered? Any problem? Look at that. The genetic code was discovered because the triplet code which was theoretically proved. The genetic code, behavior of the genetic code. What is the behavior of the genetic code? It was theoretically proved by George Gamow, a scientist, Russian scientist. He gave some theoretical evidence about the nature of the genetic code. George Gamow, he has formed a club. George Gamow, he formed a club. In that club, Crick and Brenner was the member. There was a discussion. It's like a Facebook club. They discussed what is the nature of the genetic code. Okay, what is the genetic nature code? But scientific evidence about the genetic code was given after 1965. The scientific evidence of the nature of the genetic code was given in 1965. those scientific experiments are not necessary in this course. But you remember that Francis Crick was the responsible person for this kind of genetic code. So what is the nature of the genetic code? The first characteristics of the genetic code is it is triplet. Genetic code is triplet. So you know that there are 4 bases A, T, G, C. If the genetic code is made up of 1 basis, how many amino acids will get it? You know that there are 20 amino acids. You know that there are 20 amino acids. If only 4 base is there, if there are only 4 base means A, T, G, C or A, U, G, C, whatever it may be. If there are 4 bases, that means 4 amino acids, still will be shortfall of only 4 codons will be there. That means 20 amino acids with 4 codons, it is impossible, is not it? Then what will happen? Suppose genetic code is comprised of 2 codons, what will happen? 2 bases, what will happen? So if genetic code is made up of 2 bases, how many combinations we will get it? If the genetic code is doublet, we will get only 16 combinations. You can count it. If G can, that means look at that. If genetic code is made up of 2 bases, that means we are having altogether 16 genetic code. Am I right? So 16 genetic code means we are having 20 amino acids. So it was the theoretical discussion at that time. It was the theoretical discussion at that time. That means what happened? Single base is not sufficient to become a genetic code because we are shortfall of 16 bases. 2 base is not sufficient to specify genetic code for 20 amino acids. Still we are the shortfall of 4 bases, okay? 4 codons. So what was the hypothesis was that at that time? If there are 3 bases at a time, what will happen? Then it is possible to have 64 genetic codes. If there are 3 bases at a time to become a genetic code, that means the maximum number of, in all permutations, we will having 64 codons. We are having 64 codons. That means 20 amino acids are there. That means 44 is extra. Am I right? Based on this argument, it was hypothesized that in 1963 and 64 that genetic code is triplet. Are you getting my point? That means to become a genetic code for 20 amino acids, there must be 3 bases to become a genetic code. Am I right? Is it any problem? Is it clear? That means this is the process. That means how many is become redundant? 44 is still extra. 44 is still extra. That means minimum 3 base should be there. The hypothesis was that to become a genetic code for an amino acid, minimum number of base should be 3. Minimum number of base should be 3. Suppose if there are 4 bases at a time, so that means if there are 4 bases at a time, it will have 256. If there are 5 bases at a time for a genetic code, it becomes 1024. So it will keep on increasing. So by looking at this type of arguments, finally it was decided that, theoretically it was decided that genetic code, minimum number of bases to become a genetic code should be 3. With 44 extra bases. Is it clear? This is the theoretical argument. Finally, scientifically it has been proved that in due course of time, it has been proved that yes, genetic code is triplet. Triplet means made up of 3 bases. So this is the first characteristic of a genetic code. Okay, so genetic code is triplet. Why genetic code is triplet? This is the explanation, theoretical explanation at that time. Okay, next is genetic code is non-overlapping and commaless. Genetic codes are non-overlapping and commaless. Genetic codes are non-overlapping and commaless. Non-overlapping means a particular base is not shared by 2 genetic codes at a time. A particular base is not shared by 2 genetic codes at a time. And commaless means there is no punctuation mark, there is no comma, there is no full stop. Of course, full stop is there. There is no comma. Comma means between the base, 1 genetic code is there, then there is no extra base. If what will be a comma? In a genetic code, genetic code is made up of certain bases. Genetic codes are made up of certain bases. Okay, look at this. What is the genetic code? This is genetic code. Suppose G A T C A G T G A. So how many genetic codes are here? So if the genetic code is triplet, that means we are having 1, 2, 3 genetic codes. So if there is any comma or punctuations here, can I have any comma here? Because if you look at the DNA structure, there is no structure to say that there is a comma. Because the DNA structures, these are the bases, they are joined together. In between the bases, there is no comma or punctuation marks. That means any punctuations you are requiring here, it must be a base. You must require a base. Okay, you must require a base. But in between, suppose this is a codon here. So in between bases, there is no chemical structure to serve as template, to serve as punctuation mark. So it must be a base. Suppose a particular base is there. If extra base 1 or 2, minimum 1 base, if any extra base tries to serve as punctuation marks like comma, if any minimum 1 base tries to serve as extra base, what will happen? Genetic code that ultimately this look at that 3 9 plus another 3 12. Minimum, if one base is a comma or full stop, you will have this much of bases. So that means what happens? Then if any base act as a punctuation mark, total distance will be increasing. Length of the DNA will increase or not, but length is remaining constant in species, within the species or species. So ultimately it is disadvantageous for an organisms. So that is why genetic codes are commaless. Okay, punctuation. Look at an overlapping code. What is an overlapping code? Look at that. First base, genetic code is triplet. GTC is there, the first codon. Then in the second codon, second and third base of the first genetic code is shared. Am I right? In the first base, look at this. GTC is there. For the second code, first base and second base of the first code is shared with one extra base. These are second codons. So there is a sharing of the bases in the subsequent codons. So there is a sharing in the subsequent codons. So when there is a sharing of the bases between bases of codons, what will happen? It is a total disadvantageous situation. It is a total disadvantageous situation for the organisms. Okay, it will be total disadvantage because information read during translation process. Because codons are fixed for amino acids. The genetic codons are fixed for the amino acids. So when there is sharing of the bases, informations will be wrong information will be given. So many codons will be there for a particular amino acid because 20 amino acids are fixed. So if there is a codon sharing like that, so many information, so many codons, that means information generated will be complicated. Again, look at that. So there are different ways to look at the reading frame. Okay, so look at the reading frame. What is reading frame? What is a reading frame? This is a reading frame. How many glasses I have in my reading frame? 2 glasses. For a reading frame, for me the reading frame is comprised of 2 glasses. Like that, in a genetic code, reading frame is comprised of 3 bases. A reading frame in a genetic code is comprised of 3 bases. My specs contain 2 glasses. 2 glasses are reading frame. So genetic codes has a reading frame of 3. Okay, a reading frame of 3 bases. So when there is a sharing of the reading frame, so this type of makes overlapping codons, reading frame will be problematic. So when there is a problem in reading frame, if there is a problem in my reading frame, what will happen? If there is a problem in my reading frame, what will happen? I cannot see it. All information what I read, it will be problematic for me. Like that, in the biology also, sharing of the bases in the genetic codons, it will be problematic. It is non-ideal. So that is why remember that it is non-ideal. So ideally genetic codons are commaless and without any overlapping. Okay. The next question is, it is unambiguous. What is unambiguous? What is unambiguous? What is unambiguous? So unambiguous means a single codon is there. Ambiguity means a single codon for many amino acids. Ambiguity means there one codon codes for many amino acids, but codons are unambiguous. A single codon, it will specify only one codons. A single codon, it will specify only one codons. Okay, a single codons will be specify only one amino acids. Exceptions are there. It is the exceptions are there. There are three exceptions. So AUG, it codes for formyl methionine in eukaryotes, and in the beginning of the codons, in the beginning of the protein synthesis, AUG codes for formyl methionine, then AUG codes for methionine other sites. Okay, methionine other sites. So look at this. During the protein synthesis, AU is a start codon. In the beginning, the first codon will be AUG. So in the beginning of the translation process, the AUG codes for formyl methionine. But in subsequent places in the genetic code, AUG codes for methionine. Okay. Then UUU, it's code for phenylalanine. Look at the third sentence. UUU codes for phenylalanine. So what is happening? In presence of certain antibiotic, same codons, this UUU, it's instead of coding for phenylalanine, it codes for something others. It codes for something other when in presence of streptomycin. In presence of streptomycins, same genetic codes for different amino acids. So this is a practical implication in our life. This has a practical implications in our life. Why? Look at that. Antibiotic is there. So if we take excessive dose of antibiotics, if I take excessive dose of antibiotics, what will happen? Those antibiotics will interfere in the genetic code behaving system. Look at this. If we take excessive antibiotics, instead of coding for a particular amino acids, it codes for different amino acids. So it leads to problem in our case. So that is why one of the reasons for reducing the antibiotic is, this is one of the reasons. Are you getting my point? Is it clear? Particular codon is for mean for something else, but in presence of the antibiotics, so that codon will code for different amino acids. So if there is a different amino acids, what will happen? It will be wrong formation of the protein. Proteins are again enzymes will be defective proteins are produced. So this is the problem. Okay. Then there are few nonsense codons are there. These are the few nonsense codons. Okay. So there is only, these are the few nonsense codons. These are codes for nothing. So remember that this is your, look at my, this is my initiation codon. This is the initiation codon. These what is nonsense codon? These three genetic code does not code for any amino acids. What is nonsense codons? These three amino acids does not code for any amino acids. Okay. So these are referred as termination codons. This is UAA. This is UAA, UGA, and UAG and UGA. Remember, it is wrong. UAA, UGA, and UAG. Three codons are there. These are three referred as nonsense codons. This is, you should read it UAA, UAA, UGA, and these are nonsense codons. Why it is nonsense codons? These three genetic codes does not code for any amino acids. So look at this. What is this tree? These three. What this is one AUG. It is the initiation codons. These initiation codon code for formyl methionine. I repeat, AUG is the initiation codon. AUG is the initiation codon. Its code for formyl methionine. So this codes for formyl methionine. Okay, AUG is the initiation codon. It codes for formyl methionine. Okay, at the beginning, but the same codon in other place it will codes for methionine. Remember this one. So these three codons, these are nonsense codons. Why? This does not code any. So the ultimately does not code any amino acids. That means what does it mean is that these three codons serve as full stop. These three codons serve as full stop. So, okay, in a polypeptide sense, okay, in amino acid sense, okay, they serve as full stop. Again, codons are universals. That means a codes for same amino acids in all organisms. Universals, codes are universals. What does it mean? A codes for universal means what happened? These genetic codes, same genetic code in me, you, plant, microbes, same. There are certain exceptions are there. The universal codes use a stop codon, but human mitochondria, the 4 codons, exceptions are there. In life, always exceptions are there. So again, except in human mitochondria, you don't. Except in human mitochondria, all codes are same. So UGA, it is a stop codon in general, but at the same time, UGA, it codes for tryptophan in human mitochondria. AGA, it's arginine, it is a stop codon in human mitochondria. AGG, it is arginine, it is a stop codon there in my human mitochondria. AUA, this is one of the amino acids, this is again methionine here. So these are the 3-4 exceptions are there. Only that is only in human mitochondria. These are the 3 exceptions only in human mitochondria. Okay, these are the 3 exceptions in human mitochondria. So look at that. Ultimately, what happened? To when genetic code was deciphered, Nirenberg, Mathai, Watson and Crick. So ultimately, all were done by scientists like Nirenberg, Mathai, etc. They did all chemical experiment. This chemical experiment, they proved that they ultimately finally produced. This is the dictionary. How many words are there in dictionary? How many words are there? How many words are there in this dictionary? 161, 62, 63, 64. 64 are there. In those 64 genetic, this is the coding dictionary. There are 3 codons are there, which does not code for any amino acids. AUG there. Where is AUG? If you see AUG, it is start codons. It codes for formyl methionine. In other place, it is called methionine. Except this, there are some. If you look at this coding dictionary, what you will see that there are some codons. So that means what my point is that some amino acids are coded by more than 1 codons. Some means most of the amino acids. There are 20 amino acids. Some of the amino acids are coded by more than 1 genetic code. Except two. Okay. So this is. So look at this. So what does it mean is that? So it means that there is degeneracy of the genetic code. What is degeneracy? Amino acids are coded by more than 1 codons. There are more than 1 codon for many amino acids. Okay, so this is called degeneracy. Again, codons specifying same amino acids are called synonyms. My important point I want to emphasize is that there is degeneracy of the genetic code. What is degeneracy? Remember that for a genetic code, for an amino acid, there are more than 1 genetic code. What is degeneracy? For an amino acid, for 1 amino acid, there are more than 1 genetic code. Why? 20 amino acids, 64 genetic code. Obviously, that means more than 1 genetic code should be there for an amino acid. So what is why there is degeneracy of genetic code? what are the reasons for degeneracy of the genetic code. So there are, look at this, most of the amino acids are coded by more than one codon. That means code is redundant. Okay. Look at this degeneracy. Look at the example. There are, for serine, the particular amino acids for serine, how many amino acids are there? 6 codons are there for a particular amino acid. There are 6 codons for amino acids. So that means more than one codon is that out of 20 amino acids. Remember, out of 20 amino acids, 18 amino acids are coded by more than one. Out of 20 amino acids, 18 are coded by more than one codons. Okay. Exceptions are there, methionine and tryptophan. Methionine and tryptophan has only one codon. Methionine and tryptophan has only one codon. That means these two gone, then 18 of the genetic code, 18 amino acids are coded by more than what? More than one codons. So the next question is, why there is degeneration of the codon? Why this is called degeneracy? What is the reason of degeneracy? What is you must have to understand what is degeneracy of the genetic code. What is degeneracy of the genetic code? Degeneracy of the genetic code is, what is degeneracy of the genetic code? Degeneracy of the genetic code means one amino acid is coded by more than one codons. When there is more than one codon present for a particular amino acid, that situation is referred as degeneracy of the genetic code. So reasons are, some tRNA could recognize more than one different codons. So there are, that means 61 codons are there. A particular tRNA molecule, you already know that. What is the function of tRNA molecule? Can you tell me what is the function of tRNA molecule? You remember, you know that tRNA carries the amino acid to ribosomes. Okay, so they carry particular, each tRNA carry particular amino acids in recognition to a particular codon present in the RNA molecules in the ribosomes. So there are certain tRNA molecules which can recognize more than one genetic code. Again, some point is that this is one of the reasons. The second point is that there is an Wobble feature at the base of the 5' anticodons, and that is ability of the codon base pair with different codons. So what is a Wobble feature? So you know that this is a tRNA molecule. tRNA molecule is there. This tRNA molecule, it is a, this tRNA molecule is again it is an mRNA. This is a genetic code. In the RNA molecule, it is a genetic code. Again, this tRNA molecule, it carries a particular amino acid here. This particular tRNA molecule carries an amino acid. So that means if it is 5', okay. So for this codon, look at the codon. What will be the anticodon here? In the tRNA molecule, what will be the anticodon here? A pairs with U, C. So this is the codon, anticodons. This is the anticodon of the tRNA molecule. It has UAC, and this codon anticodon pairing, so when it is matched, there will be a particular amino acids during protein synthesis. So what is Wobble hypothesis? That the base position in the end, in the 5' end, this is the 5' end of the tRNA molecule. The base position in the 5' end of the tRNA molecule, there is an Wobble feature. Okay, Wobble feature means what? It can recognize, there may be one particular base, it can recognize more than 3 bases here. So what will happen? Or in other words, the first two base of the genetic code, look at me, the first two base of the genetic code is fixed. First two base of the genetic code is fixed. But the third position, this base may change. This base may change. It may be either G, or it may be T, or it may be U, like this. Any base may be there. Any, that means what happened? The change in the 3' base of a genetic code. Look at this. The change in 3' end base of a genetic code can be recognized by one particular tRNA molecule with a particular sequence. Are you getting my point? It may be, or initially it may be AUG, it may be AUU, AUC. So what is that? You have 3 amino acids, 3 genetic code. Okay. When there is an Wobble feature, that means look at this. The change only in the 3' end of the bases. Okay. This is 5', this is 5', this is 3'. So only in 3' end, base has been changed. So any change in the 3' end of the base can be recognized by one tRNA molecule. One tRNA molecule. This tRNA molecule, suppose having UAC. Suppose this is the tRNA molecule. Example, there is a change in the 3' end of the base. That means you have 3 genetic code. That 3 genetic code can be recognized when there is an Wobble feature in the 5' end or 3' end. That means 5' end base of a tRNA molecule can pair with 3 genetic code. Suppose this is a tRNA molecule UAC. Example, this UAC carries for an amino acid like arginine. Okay, this is arginine. This is a tRNA molecule. It carries a base for arginine. It is fixed UAC, but there is a sense pattern of the bases in the 3 genetic code. First 2 is fixed, third base of a genetic code sense. That sense will be ignored by the tRNA molecule to carry a particular amino acids. Are you getting my point? So that is how for a particular amino acid there will be 3 genetic base. Is it clear? Suppose first 2, this is mRNA molecule, this is the tRNA molecule. tRNA molecule is fixed. It is UAC. It carries a particular amino acid called arginine. In the 3 codons, these 3 codons, they differ only for the third bases. What will happen? The particular amino acid carrying tRNA molecule, it will recognize all the 3 codons. So that is known as Wobble feature. Is it clear? That is known as Wobble feature here. What I mean is it is here. The Wobble feature, the base at the 5' end of the anticodon has the ability of base pair with more than one positions. It is called Wobble position. The third position is Wobble. Third position is Wobble position. So this is wobbling. The third position is changing. It is known as Wobble. Don't try to copy here. It is totally useless unless you read the book. What is Wobble? What is Wobble is the third position of the base genetic code is changing. It is Wobble means some alternate bases are coming. So that alteration of the bases in the 3' end, it can be recognized by only one tRNA molecule carrying a particular amino acids. So this is known as Wobble hypothesis or Wobble positions. Look at this. So what it was, Wobble hypothesis was proposed in 1966 by Francis Crick. So it states that the base in the 5' anticodon is not specific as the other two, or base position in the 3' end. This is important. What I explained is that this sentence is important. What is Wobble hypothesis? That the pairing of the third position bases of the codon is ambiguous. First part is, forget about the first part. Look at this. What I explained here. The base pairing of the third position of the base of the codon is ambiguous. Can be ambiguous. Ambiguity is there. Third position. This is the third position. third position base is ambiguous. Third position base is ambiguous, which is known as Wobble hypothesis. This ambiguity in the base, third position base is still recognized by one tRNA molecule. Are you getting my point? Third position base is ambiguous and it varies according to the nuclear present in these positions. So it is ambiguous. Third position is ambiguous. G, U, C. Okay. This 3 genetic code is recognized by one tRNA molecule. So that is how this is called as Wobble hypothesis. Look at that. Look at that. Suppose in and base U or C is there. U or C is there. Either U in this position, either U or C is there. When there is U or C, then any amino acid, any tRNA molecule carrying 5 in the 5' and G, it can recognize the same codons. Are you getting my point? Suppose G is here in my codon. Anticodon, it is G in the 5' end. So if it is U or C, then it will recognize. This is a synthetic base, inosine. This is a synthetic base. This is in experiment it has been proved that this is a synthetic base not found in normal DNA. Inosine, INO, SINE. The synthetic base, when there is in the synthetic base is present in I, it can recognize 3 genetic codes with having 3. Look at the example now. Look at this example. This is experiment. This is a tRNA molecule structure, chemical structure. It carries the amino acid alanine. So in the 5' end, this is the 5' end of the codon, anticodon. So in the 5' end, I is there, inosine. Okay, I is there. It is a chemically synthesized tRNA molecule. I is not present. So look at that. When I is present in the 5' end, it can recognize any codon with GCU, GCC, CGA. What is the difference between 3 codons? What is the difference between 3 codons? There are 3 codons. Only difference is that in the 3' end, the last base of the genetic code changes. Last base is U, C and A. There is Wobble feature in the last base of the DNA. So any change in the last 3 bases can be recognized by one amino acid. That means this tRNA molecule, it is same. This tRNA molecule is same. It is having CGI. I is a synthetic base. That means for one gene, that is this is carrying alanine. This is one amino acid known as alanine. This tRNA molecule can recognize all the 3 genetic code. So this is called Wobble feature of the genetic code. Because of this Wobble nature, there are more than one amino acids, more than one genetic code for one amino acids. Are you getting my point? How it is there? How there is more than one genetic code for a particular amino acids? It has been said that because of the Wobble feature, ambiguity in the base pairing in the 3' end, ambiguity of the base in the 3' end, that will be recognized by one tRNA molecule. This is called Wobble feature. So is it problematic for you? Are you getting my point? Is it clear? That means in the 3rd position of the genetic code, in the 3' end, there is a change in the base. In the 3rd position of the base, there is a change. So that change is ignored by the tRNA molecule carrying the particular amino acids. Is it clear? So that is known as Wobble feature of the genetic code. So this was proposed by Francis Crick in 1966. Okay, this is already I explained. So it occurs in the 3' end of the codon or 5' end in the anticodon. This is what I explained. Wobble feature ambiguity occur in the 3' end of the codon or in the 5' end in the codons. Okay, so ultimately, this when there is degeneration of the genetic code, it translates the same amino acids. Okay, same amino acids. So because of that reasons, you are having more than one genetic code for a particular amino acid. So with this I stop my today's lecture.